Hello everyone, myself Nishat Supugde and I am your instructor and resource person for this 4-week international level MOOC on Bioinformatics Essential, Databases, Sequence Alignment and Ethics. This MOOC is brought to you by Department of Microbiology, KK Arts, Commerce Science and Computer Science College, Saraswati Nagar, Nashik in collaboration with Bioinformatics Services. I welcome you all in the second week and module 2.1 of this particular MOOC. So, in this module, we have named that this module is the biological sequence data, formats, pre-processing, and functional annotations. So, currently in this week, we'll look for the understanding biological sequence formats. Then we'll look on data pre-processing. Then we'll explore about the databases, and then in the last we'll see about UniProt KB and the demonstrations based on that UniProt KB and the gene ontologies. So from biological material to digital data, how it goes. Modern biology generates enormous quantities of digital information. Okay, you can take any biological sample such as a blood, tissue, or bacterial culture, or a plant specimen can eventually you can take, and that you can produce DNA, RNA, or protein sequence from it. You can do a sequencing, and that sequence will give you digital sequence data, and if you upload that data you can upload that data in the databases, and you will do the biological interpretation based on that particular data. So the bioinformatics here provides you the computational methods to required to store, organize, analyze, and interpret this data. Now, what is a sequence format? Okay, a sequence format is a standardized way of representing biological sequence information in a computer readable form. Okay, a one format determines how the sequence itself is represented, how sequence identifiers are stored, how biological annotations are represented, how additional information is organized, and different formats were developed because different biological questions require different types of information. So far we can say that a sequence is the biological information, and a sequence format is the way that information is organized and stored. Now, why these standard formats are necessary? Now imagine one you are receiving laboratory results, okay, and every laboratory has a different arrangement of the report. Okay, one report might place patient's name at the top, another might place it at the bottom. Now there is no matter, but another might use abbreviations that are not defined. So automated interpretation would become difficult if we see all this scenario. So if there is no standard formats are available, then the same problem will occur with the biological data. So these standardized formats will allow databases to exchange the informations. Okay, believe me, all the databases that we have visited, and the many databases we have not visited, some have visited, some have not visited, but that databases are sharing the information in between them. So databases exchange means the standardized format allows databases to exchange the information, softwares to read the sequences, researchers to share data sets, computational pipelines to process the data, and results to be reproduced. So these standardized formats will allow these five things. So key principle is standardization enables interreportability. Okay, so you can report data, you can reproduce the results if standardized formats are available. So FASTA, GenBank, EMBL, GFF, GTF, FastQ, PDB are the some biological sequence formats that will look whatever are necessary at this foundational level, that will look very closely. So here, so far, important distinction is not every biological file containing nucleotide letters is simply a sequence file. Okay. Some formats also describe the quality of the sequence, genomic coordinates of the sequence, variants of the sequence, functional features, alignment information. All these informations are stored in the sequence file. So it is based on what kind of data you want, what kind of process you are doing on that. Thank you. So this is for the module 2.1. In the next module, we'll look for the more and more deeper knowledge of this particular thing. Thank you.